Oh, a bit depleted today. A lot of people have gone off on holiday already, have they? Anyway, so this is the second of my two lectures. In this one, I'm going to take you into areas of cutting-edge research, which is still a long way from actually coming into routine use. But nonetheless, I think you should be looking at the horizon and seeing what's coming rather than just focus on what we do routinely today. I just checked the stuff is up on Moodle. I hope somebody managed to find the YouTube video. Did you find the YouTube video of last week's lecture? I'm doing the same this week as well. So in a day or two it will be up there. So just to refresh your memory, in the last lecture we spoke about using culture and then moving on from there to making libraries and sequencing DNA from single colony cultures. Today I'm going to look at what happens if we try and just get rid of culture entirely and move to culture-independent approaches, sequence-based approaches for identifying and characterizing pathogens. So my starting point for this is if we look at diagnostic microbiology, particularly at diagnostic bacteriology, we're kind of stuck in a time warp here because we're still relying on techniques in the 21st century that were devised in the 1880s, in the late 19th century. So we still rely on the Gram stain and looking down the microscope at bacteria. And Gram showed us how to do that in 1884. We still rely on methods that Robert Koch pioneered in 1881, where he showed that you could grow bacteria as colonies on solid media. And I think the question is, shouldn't we be moving on from here? Shouldn't we be actually looking for new ways of working now we're in the second decade of the 21st century? Now, before we go into this in more detail, I'll just take a couple of slides as a side sort of angle on this, just to remind you that actually microbiology is, even applied to humans, is more than just pathogens and infections. And we have this human microbiota. We can be viewed as superorganisms built from human and microbial cells. Julian Davies in Canada made this kind of provocative statement that humans can be defined as large, highly complex microbial communities attached to some relatively uninteresting organic matter. That's obviously a bacteriologist, chauvinist way of looking at the world. But there is a lot of interest in this human microbiota. People say it's a multicellular organ, in effect, as complex as the liver. And in terms of its genome, the microbiome, as it's called, which is all those organisms that are in the microbiota, plus their genes and genomes, that comes out as over 100 times more genes than your so-called human genome. For those of you who are wondering where's the second genome is obviously the mitochondrial genome, which is in fact a bacterial genome as well. There are a number of interesting assumptions or realizations that have come into microbiology as we've basically looked at microbial ecology and environmental microbiology and imported these ideas across. And so one idea is that although there are these large numbers of organisms that live on us, many of them actually can't be cultured in the way that Koch wants us to culture things, in growing them in the lab, getting single colonies. And so there's been a lot of interest in using molecular methods, culture-independent approaches, just to perform a census of what organisms live there and characterize the composition of that microbiota in health and in disease. And one of the pioneers in this field is a guy called Scott Dowd in the US, and he has been using culture-independent approaches, say, to wound infections. And he claims that you actually, if you look in the round, you get better management of the wound infection when you take into account the things you pick up with the culture-independent approaches than you would if you just looked at what you find in culture. Here's one of his papers. He's had a couple of papers like this, but here is one where he said that basically that there was an improved outcome in wound care when you looked at the molecular approaches, the culture-independent approaches. Now, just to stay for another couple of minutes on this human microbiota, We actually often think of bacteria as our enemies, you know, and in medical microbiology think of them as the enemy, pathogens and so forth. But in fact, this human microbiota that we all carry brings many beneficial effects. There's this thing called colonization resistance, where it competes with pathogens and prevents them from getting a foothold on body surfaces. It's involved in normal development, normal homeostasis of the bowel and other factors. It's involved in the assimilation and production of nutrients, and we have complex polysaccharides that are broken down, and it's been argued that about 10% of your dietary intake is actually cycled through these bacteria. We're also clear, though, that the human microbiota is implicated in disease. So some of these so-called commensal organisms can cause disease. So Strep mutans, which lives in your mouth, causes dental caries when you spray sugar all over your teeth. Streptococcus pneumoniae can come out of the upper airways and infect the middle ear and the sinuses and so forth. And if your gut microbiota escapes to abnormal sites, it can cause disease. And we recognise also that many organisms that are not normally considered pathogenic, in the modern healthcare context, can actually be pathogenic. So if we have immunocompromised individuals, or we have lots of foreign material, plastic and metal and so forth, some of these organisms, like Staphylococcus epidermidis, that lives on your skin, can actually become a pathogen. Now, there's been a great deal of interest also, though, in the idea that it's not as simple as headline pathogens causing disease. It's all to do with interactions between the host and the microbiota. And people are starting to say, well, actually, the human microbiota actually plays a role in well-being and so forth, well beyond just its influence on infection. One idea is this hygiene hypothesis, so that as we stop having exposure with many components in the environment, microbial components in the environment, we become more susceptible to, say, allergic conditions, asthma and eczema and so forth. It's clear also that, the gut microbiota, which is the largest of these microbiotas, has been implicated in the pathogenesis of bowel cancer, inflammatory bowel disease, diabetes and so forth. And the really hot ticket at the moment is the role the gut microbiota plays in the balance between leanness and obesity and weight homeostasis in general. Now, I'm not sure, I think David Honeybourne probably spoke to you a little bit about the problems that we have sometimes with cystic fibrosis, and this is an example where people are now starting to think, well, actually, when we look at what we can grow in the Koch paradigm as the headline pathogen, that's not enough to explain what's going on. And there are some people in this discipline, in this field, that are actually saying, well, actually, we have to look at the whole microbial community, and if you like, treat that as a pathogenic entity, and it's not enough just to look at one or two organisms. I haven't put the word down, but there's this very fashionable word called dysbiosis to describe where that community, a microbial community, comes out of kilter and causes disease. And I just already said this point, but just to make the point again, the inflammatory bowel disease, like Crohn's disease. Crohn's disease is not an infection in the classical sense, but it's clear that the microbiota is influencing the outcome of disease, and in some degree this is an abnormal response to that microbiota that is causing the disease. And just to give you one, this is a bit rather old paper now, but this is one of the ones that started off this whole interest in the gut microbiota and things that aren't infections. Here was one where they actually defined an obesity-associated gut microbiome, which was able to harvest more energy and present it to the host. Now, that was a little bit of a diversion just to contextualise some of the work that goes on out there. There's a lot of papers nowadays looking at these culture-independent approaches to studying the microbiota, and as we'll see in a short while also, there is the potential for using them in diagnosis. Now, some of you may have heard of 16S ribosomal RNA. This is a universal component of all life forms. All organisms that have a ribosome will have a 16S. The actual homologous molecule in eukaryotes is called the 18S, but it is effectively the same molecule. 16S is the term applied to everything that occurs in bacteria. And bacterial taxonomists and ecologists have used this molecule for quite a few years now. A guy called Carl Woese, who died a few months ago, was the pioneer in this. And the reason was because it's universal, it's present in all organisms, but it also has this characteristic where there are parts of the molecule which are conserved and other parts which are variable between different species, between different taxa. And so you can hoik out the molecule using the conserved bits. So in fact, you can amplify the whole 16S gene from beginning to end using some conserved primers. But then you can sequence through the bits that are not conserved, and this gives you a kind of barcode for identifying organisms. And there is a massive knowledge base out there now, thousands and thousands of 16S sequences associated with known taxonomic groups, typically with species. And so you can use this molecule as a diagnostic tool and as a tool in microbial ecology. It has sometimes been called the one-eyed king. There was a paper that reviewed the problems with it. That's a reference, I think it's H.G. Wells, in The Kingdom of the Blind, the one-eyed man is king. This is still kind of king in terms of microbial ecology, but there are problems. There are problems in that not all cells lyse equally well. So if you've got a population of cells and you try and extract DNA, some of those cells will lyse easily. E. coli falls apart quite easily. But say Mycobacterium tuberculosis, you have to work really hard to get any DNA out of it. And so that creates a bias there. Although we talk about these conserved regions and universal primers, in fact there are issues there. There are some groups where the primers don't work as well as they might. And in the PCR, you usually do this by PCR, you amplify out the molecule with the conserved primers. It's a competitive process, and so there may be some molecules get amplified more than others, and so what you get out in terms of the abundance of different sequences might not actually reflect what was going into the process at the beginning. Here's a paper from a group in Hong Kong who I've had some dealings with in the past, where they actually reviewed the use of 16S gene sequencing for bacterial identification. And so what you can do in the clinical lab is you've got an organism growing in front of you, you don't know what it is, you can just do a PCR to get its 16S gene and then you can sequence it. And this is what they did, and they summarise. They say that basically it is a very useful technique in this kind of a setting. But as I say, I'm not, 16S, you see there's a lot of literature out there, but it's not something that we particularly in my group are interested in doing much more in the future. What we're interested in doing is saying, well, actually, can we move beyond these kind of molecular diagnostic approaches where you amplify or capture things and actually go towards what we might call diagnostic metagenomics? So what does that involve? Well, basically, it means that you just extract the DNA from your sample en masse, all the DNA molecules in there. Together, they are then sequenced, and you get a metagenome. So a metagenome is just a collection of individual genomes joined together, jumbled up, if you like. It's a bit like taking several different jigsaw puzzles that you might have at home and just taking all the pieces and mixing them all together into one big jigsaw puzzle. But the interesting thing is that we may then be able to get two pathogen genomes from that metagenome. And this may be a very simple way of doing things. So the advantage is it's very easy. You just go and extract the DNA and you sequence it. There's none of this business, oh, I've got to grow this organism in CO2, this one anaerobically. I've got to put that organism on a special selective medium or whatever. You could just do the whole thing through sequencing. That's the dream anyway. Of course, the reason that this is research and not routine practice is there are some problems. At the moment, it's too expensive by an order of magnitude or more for routine use, and it is rather technically demanding. But nonetheless, I think this is the direction of travel. Now, the first hints that this was an interesting approach actually came from virology. So as a bacteriologist, we bacteriologists are always going, oh, we're doing wonderful, exciting new things, sequencing genomes, sequencing metagenomes and whatever. We kind of forget that the virologists have got there before us usually because they're dealing with genomes that are 100 times smaller than what we deal with. But here are 3 papers that came out in the last few years where they used sequencing, unbiased, just sequence all the stuff in front of them as a way of finding the pathogen. So these are, in a broad sense, you could call them metagenomics. They're actually more like, you should call them metatranscriptomics, if you like, because these are all RNA viruses that have been discovered by taking tissue samples, extracting RNA from them, and then sequencing all of the RNA. So in that situation, most of the RNA you're sequencing is actually human in origin. But if you go deep enough, you can find reads that come from the virus, and that's what's happened in each of these cases here. So a new arenavirus in the case of transplant-associated diseases in intensive care units, and 2 new viral hemorrhagic fever viruses. Now, to go back to the story I told you last week, you remember that we had this interesting study where we looked at the German STEC genome, and we did all this open source genomics, and we felt very pleased with ourselves. You know, we were giving ourselves a pat on the back here. We met our German collaborators and had some champagne, and we said, well, what are we going to do now? Can we do some interesting studies in the future? And we said, well, what do we have in the UK in our group? Well, we have we can do a lot of sequencing. We're good at that, and we can do bioinformatics. We're good at analysing sequencing. And the German guys, they said, well, actually, we've got a whole freezer full of fecal samples from the outbreak. We've got hundreds of them. That's a unique selling point, isn't it? A unique resource. And so we got together and we said, well, all right, we'll just sequence your fecal samples and see what we can find out. So the idea was, when we did our first study, we went from the fecal samples through to a plate, we picked the colonies and then sequenced it to get that genome. Could we go directly from the fecal samples to getting sequence that would allow us to identify, characterize the outbreak strain and maybe other things as well? So they sent us some samples, and the very first sample we looked at, we were very pleased with what we found. It was known to have a high abundance of the sugar toxin producing E. coli. And what happened was that Nick Lohman in my group at the time, he took the metagenome, so we sequenced it on a benchtop instrument, the MiSeq, 1 sample, 1 run on the MiSeq. He aligned all of the sequences from that metagenome against the reference genome that we already had for the outbreak strain. And it's a little bit annoying the way this is being projected, because these coordinates here, these are just the coordinates in the genome. To put them in scientific convention, that doesn't really help. But basically, this is 5 million, 4 million, 3 million. These are the positions along the genome. And these little dots here are where there is a match in the reads from the metagenome. And what we end up getting here is, for each sequence in that genome, we're getting between 20 and 30-fold coverage. So there are at least 20 different sequences in the metagenome aligning. So that's about what you'd expect to get if you were trying to sequence this genome from a colony. That's the kind of coverage we aim for. So we're very pleased with that. You can see that there is a sort of skew in this alignment here, and that's because when you have rapidly growing cells, the origin of replication is actually overrepresented in the population, whereas the bit just before you get the origin of replication as you're going around is underrepresented. Now, the other thing that we found, interestingly, was that there was this little upswing here of sequences at a particular position, and that position turned out to correspond to the prophage, the bacterial virus that had integrated into the chromosome that carries the genes for the sugar toxin. And it was known that the sugar toxin was encoded by this phage. And what we interpreted this finding to mean was that we were not just detecting DNA coming out of the chromosome of bacterial cells, but we were probably also detecting DNA from free phage particles that were actually present in that stool sample. And it's known that what happens is that the phage actually lyses the cells as part of its life cycle, And it's the lysis of those cells that actually releases the toxin. So it was quite satisfying to see this phage lysis, which is an essential part of the pathogenesis of infection, visible to us in this particular plot. We were also able to reverse engineer the sequence type using conventional sequence typing approaches from the genome, and we could actually say, so we know that this, just looking at the metagenome, we can say that this E. coli is sequence type 678. So then we looked at a range of samples, and what we agreed with our German collaborators was that we would look at some samples that had the STEC, they knew were STEC positive, but we'd look at some other samples that they'd isolated, that were taken at around the same time during the outbreak, patients with diarrhea, but where they didn't have STEC, but they had another pathogen instead. And so we then aligned against various pathogens, reference pathogens there, and we got these kind of plots. So for some plots, we got 1-, 2-fold coverage. Most of these, we're not seeing anything like what we saw with that first STEC sample, but nonetheless, we are getting genome-wide coverage and clearly have been able to identify different pathogens in the samples. The 3 panels at the bottom there, these are different E. coli positive samples, different depths of coverage, but in each case we see that little upswing where we get the prophage and the phage sequences there. Curiously though, the ratio of those phage-related sequences to the backbone changes, and we don't quite know why that is in different samples. In this sample over here, we see an interesting phenomenon in that as you're going along the coordinates of the STEC genome at the bottom, the outbreak strain genome, and you look upwards, you can see that there's actually 2 lines across there, if you like, as we're going through across that alignment, with different depths of coverage. So there are some things that are covered around 1,000-fold in that particular metagenome. Those are the parts of the STEC genome which are actually shared with all other E. coli's, the so-called core genome of E. coli. And then beneath that are some other sequences which form part of what we might call the accessory genome, the strain-specific sequences that belong only to that strain. And what this plot is telling us is that there are in fact 2 different E. coli, at least 2 different E. coli strains in this sample. There's the outbreak strain and there's another one. And where there is core genome from both strains, you see this much increased coverage. There's one last thing I should say before we move on from this slide, and that is that Campylobacter concisus there in the middle at the top. That actually hadn't been reported. So the other things, Clostridium difficile, Campylobacter jejuni, STEC and so forth, have been reported by the routine clinical lab. But we found this Campylobacter concisus unexpectedly in a sample that had been badged as being C. difficile positive. And we recovered far greater coverage. We did recover some C. difficile reads from that sample, but the Campylobacter concisus was present at much higher abundance. And neither myself nor Nick had heard of this particular species before. We went and did a literature search, and it turns out it has been described as a potential or an emerging diarrheal pathogen. And because it was there in more abundance than the C. difficile, it does beg the question of whether it was contributing more to the pathogenesis of diarrhea than the C. difficile, which is a kind of interesting point. And it wasn't looked for by the routine lab, so they wouldn't have ever found it. Actually, before I move on there, one other thing we found was that in some of these samples that had STEC, they also had C. difficile. They're in lower amounts, and we still don't quite know what that means. Does it mean that the C. difficile was just a bystander colonising this inflamed bowel or whatever and making use of the ecological disruptions? Or was the C. difficile actually also contributing to the diarrhea and to the disease alongside the STEC? And it's an interesting question. Now, we tried to submit this to the Journal of the American Medical Association, which is a highly prestigious journal with an impact factor of 30. And we thought, yeah, we're in with a chance here. But the vibes we got back from the editor when it was first looked at was, this is no good. because they're having a special issue on genomics, and we'd established, can we do microbial genomics? It was genomics in health, and they said, yeah, all right. But then they said, well, you've got too few samples here, because we only had 10 samples. We had 5 with STEC, 5 other things. That wasn't enough. And we were only getting 67% sensitivity in detecting the things we were looking for. So they said it's a lousy test. And another issue that was raised, I think probably by a guy called David Relman, who's a leader in the field, and I think he was the reviewer. He said that we cheated, because what we've been doing is we've been taking this metagenome, we've been aligning it against the known outbreak strain. So if you go back to the analogy I made earlier about having, taking a load of jigsaw puzzles and throwing them all together, effectively we were taking the lid of one of those jigsaw puzzles and we were using that as a way of actually pulling out the pieces. And he was saying, no, you shouldn't have done that. You should have done it as if you didn't know what the cause of this outbreak was. So you should have solved this problem without any clues, de novo, as we sometimes use in the jargon. So we realised that if we were going to get this published, we had to go deeper in our sequencing, and we had to think smarter in the way we were doing the research. We actually called up this guy, Jeff Smith, at Illumina, people who make the sequencing instruments. One of the guys in my group at the time had just left Illumina, and so we had good working relations with them. And we said, can we have a run on a much higher throughput instrument, your HiSeq 2500, just recently launched, and can we have... And he said, well, as it's you, okay, I suppose so, yes, an interesting project. And so we ended up sequencing over 40 samples on the HiSeq. And some of the samples that hadn't given us as good a result as we liked the first time round, we actually sequenced them tenfold deeper on the HiSeq than we had on the MiSeq. And it worked, basically. On the samples that weren't STEC positive, we did recover lots of interesting information. And you can see that we got millions and millions of reads, and tens of millions, nearly hundreds of millions. Well, in fact, for Campylobacter, we got over 110 million reads from the metagenome. And we were able to make inferences about the strain, its epidemiology, its pathogenic potential, and so forth that were in these samples. When we applied it to the STEC positive samples, and we now looked at a much larger number, we still were only getting about 2/3 of them where we were detecting the STEC sequences, for example, the sugar toxin sequence. Now that doesn't look so good. In fact, we kind of set ourselves up to fail in that we asked our German collaborators to give us 3 different kinds of samples, 3 sets of samples. One in which there was an abundance of STEC and it was easy to find, one where there was a modest amount, and we said, give us your hardest ones where you had the most difficulty actually getting the organism out. And it was in those that, no surprise, we couldn't actually detect by sequence either the organism very well, or at all, in fact. So we realised that if we were going to write this up for JAMA and we were going to say we've got a new test, metagenomics is a new diagnostic test, the editor and the reviewers would say, you're telling us you've got a test that costs thousands of pounds, requires state-of-the-art sequencing instruments, requires state-of-the-art bioinformatics pipelines and very clever people to do the work, and it's still only giving you 67%. What's the point of sensitivity? What's the point of that? So we had to sort of scratch our heads and think about how can we reframe this paper to make it a paper that could be published. So what we did was we said, well, instead of saying that we're going to use metagenomics to make an individual diagnosis in this stool sample, this stool sample, this stool sample, let's just see if we can use it to identify what's causing this outbreak. And that might be a better way forward. And so that's what we did. So this is sometimes called a blobogram. You see these kind of pictures. I saw a guy from Oxford getting very excited about them last week at a meeting. Basically what Nick Lohman did was he plotted the depth of coverage against the GC content, and then he plotted each of the reads in the metagenome. Actually the context of the metagenome. He did a rough and ready taxonomic assignment. And we said, well, let's see, if we're going to find this outbreak strain, it's got to be at least present in more than one sample. And so this is a kind of census of what you see in this, if you take all those samples and you say what's present in more than one sample. And as you can see, it's a huge abundance of different organisms. It's a right taxonomic mess, really. So then we said, well, let's make a simplifying assumption and say we've got this outbreak, we've got 40 stool samples in front of us. Let's say that we expect the pathogen to give us reads in at least half of those samples. We should be able to detect reads from the pathogen. So anything that's not present in at least half the samples, we can throw it out. So we did that, and we ended up with quite a bit of simplification here. And you can see the Enterobacteriales, the pinkish stuff in the middle there, which is where E. coli belongs, actually are starting to become a bit more abundant. But we're still a long way from actually having an outbreak strain genome out of this metagenome, or metagenome, if you like, because it's a composite of all the metagenomes of all the samples. So then we took another simplifying assumption. We said, well, what we want to do is we want to look for things that are not present in normal people. Now, we obviously didn't have time to go and collect 40-odd samples from normal people and metagenome sequence them, but luckily people have done this already and they're available in the public databases. So Nick went and got these sequences, subtracted anything that was similar, and that led to this massive simplification where he actually was able to identify an E. coli in that metagenome. It wasn't quite as simple as that. He had to then go out, because that only gave us the accessory genome, the parts that were strain specific. But he did some clever jiggery-pokery with a guy called Chris Quince in Glasgow, which allowed him to actually link up those sequences with the core genome of that particular strain and recover the entire outbreak strain genome from these metagenomes without any culture and without having a reference genome to align to. So we were very pleased with that, and we actually did get the paper published in JAMA as a result of that. Okay, so that's one example where we've kind of been pushing the boundaries and seeing if we can make these techniques work for us in the diagnostic arena, in the clinical microbiology arena. I'm now just going to step aside, just to one side, from clinical microbiology of normal, well, not normal, of existing people, and just focus for a little bit on ancient and historical DNA. So there's been a great deal of interest in actually getting DNA out of old material, museum material, archaeological material, and so forth. And there's been some spectacular successes. You may have heard that we managed to get Neanderthal genomes. There's a whole new branch of humans called Denisovans where they sequence the genome from a finger bone. There was the ice mummy that was found in the Tyrolean Alps, Ötzi. His genome was sequenced. Paleo-Eskimo genome's been sequenced. There is a problem, though, when you're dealing with ancient DNA. Most of the studies up till now have actually relied on amplifying things using PCR. And the problem with that is that there's a tremendous potential of carryover from the PCR itself. But the other thing is, if you're looking for human DNA in an archaeological sample, most of the human DNA will decay over a relatively quick time. And most of the human DNA around in that sample is actually going to come from the handlers, from the people who've been handling that mummy or that sample. And so there have been some spectacular failures with ancient human DNA. In the first Neanderthal shotgun sequences, they had to admit, after they had a spectacular paper in Nature and so forth, they had to come back a year or two later and say, 8% of that DNA in there was modern human handler DNA, it wasn't Neanderthal at all. And when you look at Egyptian mummies, where people are trying to genotype Tutankhamun and so forth, there's this whole schism in the field. There are these skeptics who say absolutely no way that any DNA from Tutankhamun could survive to the present day. And there are other people who say, oh no, we can routinely get DNA out of Tutankhamun and similar mummies and work out family relationships and so forth. When we look at infection, things are slightly different. There is a substantial literature showing that you can actually make retrospective diagnoses of samples that are hundreds or thousands of years old for a whole range of different diseases listed there. And in recent years, there's been some work done on paleogenomics, where you can actually fish out genome sequences of bacteria. The Yersinia pestis, the cause of the Black Death, has been sequenced, and a TB genome was fished out of some historical material from a crypt. There's less risk here, obviously, of contamination of DNA from the handler. There's still a risk of carryover when you're doing amplifications, PCRs and so forth. And there still is, in the field, there are those who believe that all the results that everyone produces are true, and there are those who say, no, I think you know, that's not TB from that sample, it's TB sequences coming out of your lab, contamination and so forth. So I got involved in this area because of these two people here, David Minikin and Helen Donoghue. They came to me saying, oh, they got an interest in ancient and historical tuberculosis. Could we get any genomes out of some samples? And I said, well, I'll have a go, but we don't want to do any PCR or anything like that. We'd just do shotgun metagenomics, diagnostic metagenomics, if you like. We didn't know what we were going to find. You could argue that Helen gave us some samples, 200-year-old samples, human samples. You could argue that there will just be too much human DNA around for you to get anything useful. It may be that whatever was there in life and caused the death of the individual would just get overgrown by bacteria and fungi and so forth that grew on the corpse after death. But we tried it out. We did the experiment. So what we actually got from Helen Donoghue was some material from a crypt in Vac in Hungary, which was opened up in 1994, and it had been sealed for over 100 years. It was used from the 18th century into the middle of the 19th century, then sealed up and just left. And for some reason, because of the atmospheric conditions or the temperature or so forth, the bodies that were in that crypt actually, instead of rotting away to skeletons, became naturally mummified. And among those remains, there were those of a woman called Teresia Hausmann, who died on Christmas Day in 1797 at the age of 28 years. Now, her chest X-ray had been taken, but was clear, but she did look very shrunken, cachectic, as we say in the jargon, and which is consistent with TB. It's a kind of wasting disease in its final stages. And a number of molecular and microbiological analyses had been performed on material from her already, suggesting actually that she did have TB and that there was quite good preservation of TB DNA in there. In fact, here's was one of the papers. In fact, what they did was they actually tried to revive the TB, and they put it into guinea pigs to see if it would grow in TB in guinea pigs. They actually stained it up, and they could see acid-fast bacilli. Here's Mark Spiegelman, who actually led the investigations, doing an endoscopic investigation of a mummy here and taking material under precautions, aseptic precautions. And here's another couple of screens. I'm just showing you papers where they had reported, yeah, they can amplify TB DNA. They can even show that TB is present, and they can even get a little bit of genotypic data to tell you what kind of TB it was, and to see that there were some differences between different mummies in the collection. So we went in and did a metagenomics run on this Teresia Hausmann, extracted some DNA from lung tissue, made a library, and we got, it wasn't a very good run actually, we only got 5.5 million reads, but nonetheless that was enough to give us something very interesting. So this is actually a reconstruction of what Teresia Hausmann looked like when she was actually deposited in the crypt, and you know, the material that we actually got to work on. So of those 5.5 million reads, less than 1% actually aligned against the human genome. So the human DNA in Teresia Hausmann had obviously decayed away fairly quickly. But 8% of them aligned against Mycobacterium tuberculosis reference strain H37RV, which actually gave us an average 32-fold coverage of the genome, which is, again, similar to what you would get if you were just sequencing TB that you were growing in the lab. Now, we scratched our heads and said, can this be real? This is too good to be true. If we'd have got like 10 or 20 reads from TB in that metagenome, I would have been happy. But to get this depth of coverage of a genome, I was absolutely astonished. But we couldn't think of any reason how this could be artifactual. We'd never grown TB, we'd never done any PCRs, and it was quite even coverage across the genome. So the plot at the top there, that shows you the coverage depth across the genome. And you can see there are a few little upswings and a few downswings. there's only really one upswing, and that's the 16S, and that's because it's finding parts of the 16S in that metagenome from other bacteria, which are also aligning against TB when we do it this way. But otherwise, it's fairly even coverage. In fact, there are these little downswings where, if you compare it with this reference strain, H6M9B, small parts of the genome are missing. And this is typical of what you see if you compare different strains of TB, parts of the gene, the so-called indels, and in fact, usually deletions, small deletions in the genome where a little bit of and using these indels, Martin Sargent, the bioinformatician who was doing the analysis on this, was able to say, well, actually it most closely does resemble this recent German strain, 7199/99, which was associated with an outbreak in Germany, and another strain called Erdmann, from this lineage known as the Harlem lineage of M. tuberculosis. But it got more complicated than that, because when Martin looked at it more carefully, he realized that there was actually evidence of two different genotypes of M. tuberculosis in that sample. And we, again, had to go and look at the literature. So how often do you get mixed infection? It turns out in areas of high endemicity, you do get up to 9% of samples actually have two strains in them. So perhaps it wasn't that surprising. And he was able to look at these deletions and say, well, actually there's two strains. One is closer to the German. They're both quite close to the German strain in terms of the grand scheme of things, but one is closer than the other. And he was even able to find, when he looked at the SNPs, the single nucleotide polymorphisms, that distinguished what we saw in the mummy from this, from a closely related reference strain. He was able to see in some cases that half of the reads had one SNP, the other half had a different residue at that position. So again, providing good evidence there was a mixture of strains in there. And also when we looked at the coverage, if you look, some places there were these deletions compared to reference where it just dropped out completely. But in other cases, the coverage doesn't drop down completely to zero, but it drops down to about 50%, which is again good evidence that there actually is a second strain in there. And we actually wrote this up and got this into the New England Journal of Medicine as a letter a few months ago, and this was our attempt to try and reconstruct the phylogeny of these strains. I haven't said, and I haven't put it on a slide, but we've actually continued this work, and we've now got over 500-fold coverage of the genomes from that particular sample, and we're doing 24 more samples right now, in fact, in the next week or two, and hopefully we'll be able to start to actually reconstruct the spread of tuberculosis in that 18th century Hungarian population. Now, just in case you think, well, that's just another little trick, it's not going to happen more. None of this is going to be more widely usable. Well, we've got into a collaboration with Liz Wellington in the School of Life Sciences, where she's interested in Microbacterium bovis, the bovine form of tuberculosis in cattle and badgers. And you're probably aware that there's been a great deal of interest in, are the badgers giving it to the cattle? If so, how so? And it's not really well understood how transmission is happening. And Liz is very interested in trying to actually get a feel for how much M. bovis is in the environment and how much is being shed by badgers in their feces. So she said, well, can you have a look at some badger feces for us? And so we did the metagenomics of badger feces. Strange things we do for a living, but there we are. We extracted DNA from badger poo. Well, in fact, Liz done that in her group. She gave us the DNA. We sequenced the metagenome, and when we did this kind of alignment against a reference, we found twofold coverage of the Microbacterium bovis in that sample without any attempt at optimizing or trying very hard indeed. So again, it shows that there's proof of principle here. Okay, I've spoken perhaps a bit too much about my own research interests, but just to show that I'm not the only person thinking this way. In fact, when Ötzi, this Tyrolean Iceman's genome, was sequenced and published back at the beginning of last year, what they found was, they could find human DNA and they could get some inferences about him as an individual, but they also found some sequences from a bacterium called Borrelia burgdorferi, which covered about 60%, about 0.6-fold coverage of the Borrelia burgdorferi genome mixed in with his genome, which led them to conclude that he had a disease called Lyme disease. And this was the first example of Lyme disease in a human sample, sort of 5,300-year-old retrospective diagnosis, if you like. In fact, just as we were getting ready to write up our metagenomic diagnosis of tuberculosis, a couple of papers appeared, kind of scooped us in a way. One showing that they looked at metagenomes of Egyptian mummies, and they claimed that they could find evidence of malaria and toxoplasma in those samples. Unfortunately, they haven't actually presented the sequence data, and I don't really think I believe it. Well, I'm not going to believe it until I see the data, but nonetheless, they've made that claim. Much more solid claim comes from this paper from Science, where Johannes Krause and others reported recovering genomes, well, a genome of Mycobacterium leprae from the metagenome of DNA extracted from a tooth. And so that shows that you can basically go back hundreds of years and get these genomes out. In terms of diagnostic genomics, so we've kind of went off. It's interesting that looking at these ancient samples is actually pushing our techniques to the limit. What if we bring those techniques back and apply them to contemporary samples? Well, a few people have been doing that. Here's a paper where they actually managed to get multiple genomes and a chlamydia trachomatis genome and genotype directly sequencing material from a vaginal swab. Again, diagnostic metagenomics. And just in the last few weeks, there's been this paper come out. It's actually not been laid out properly yet in the journal. It's still just a kind of a proof, but showing that you could actually take urine samples and extract DNA from urine samples and get genomes of the E. coli or whatever that's causing the infection. And so we've used, in the previous talk I talked about genomic approaches, now we're using metagenomic approaches, and we think that these are augmenting conventional approaches, investigations. We can find unknown unknowns, so these techniques are very nicely open-ended. There are some caveats. So our studies have been looking backwards. We've not had something come into the lab in the morning and say, right, we're going to turn this around and give you a diagnosis as quickly as possible, that sort of stuff. And it's too hard at the moment and too expensive. But as we saw in the previous talk, genome sequencing, you might say, oh, that's never going to catch on as a routine method. It has, just in the last few weeks, become routine for tuberculosis. I think if you come back in 5 years' time, at worst 10 years' time, metagenomics will have become a standard approach as well. There is, of course, one slight caveat, which is that all of this, the genomics and the metagenomics, always are relying on the fact that you can go from the genotype to phenotype. So you can say, so there's its genome, we can tell what it behaves like. And in fact, most of the time we can. But there will be some cases where a thing is resistant and we don't know why. There are no known mutations that are associated with that resistance, and we can't. And if we went just from the genome, we might not ever realise it was resistant to an antibiotic. And so there are some caveats, but we're moving forward. We're trying to do more work on that 18th century TB. We're looking at other mummified material. We're hoping to get some material from Fayum in Egypt, where you get these wonderful mummy casket portraits. So you can sort of look into the eyes of someone from 200 AD, and also you have their material, and we can do metagenomics on them. We're doing similar approaches on the gut microbiome in intensive care units. We're planning to anyway. Some of my colleagues are planning to look at what lives in Leishmania lesions and look at the metagenomics of that. And nothing to do with microbiology, but we are also doing similar ancient DNA metagenomics work on sediments from seafloors with Rob Allaby from Life Sciences as well. Okay, that's me finished. Just have to acknowledge all the various people that we've worked with. And, you know, we drink a lot of champagne. This was us drinking champagne over our JAMA paper when our German collaborators came to Warwick and had the champagne here with us. Okay, I haven't taken any questions now. There are any? Otherwise, the talk will be available on YouTube probably tomorrow. Probably won't get around to doing it today.